In class, we've learned that the order of bases in DNA determines the order of amino acids in protein and ultimately determines that protein's function. We've also seen that mutations in the DNA sequence can lead to genetic diseases. For example, in sickle cell anemia, a normal red blood cell has a nice round shape, but due to a single mutation in the DNA sequence, we can end up with a cell that has a sickle shape because of the sickle-shaped protein inside. In order to understand why a single base change could have such a profound effect on a protein's shape, we need to take a look at the structure of proteins in order to understand how that affects their function. Let's start by surveying protein diversity. One major type of protein in your body is collagen. If you look at the structure of collagen, you'll see that it's actually made up of 3 different polypeptide chains that work together. And you'd find collagen helping bind your muscles to your bones and in your facial muscles. Antibodies are a very different type of protein. These are defensive proteins. They help bind antigens on pathogens and mark them for destruction. Lactase is yet another type of protein with a quite different function. As you can see here, lactase is made up of just one polypeptide chain, all jumbled up, and its job is to be an enzyme. Lactase splits the sugar lactose into 2 monosaccharides. Insulin is a protein that acts as a hormone or a chemical messenger. Insulin's job is to lower blood glucose levels in your body. Hemoglobin is a protein we're pretty well familiar with. Hemoglobin is found in your blood cells, and the job of hemoglobin is to bind oxygen. And if you look at the structure of hemoglobin, it's a pretty complex protein. It's actually made of 4 polypeptide chains that bind together. And each of those polypeptide chains is capable of carrying 4 oxygen molecules. So if hemoglobin is fully saturated, each one is carrying 4 oxygens. And we find this, again, in red blood cells. Myoglobin is a protein related to hemoglobin. As you can see, it has a similar structure. But we don't find myoglobin in our blood, we find it in our muscles. And myoglobin is great because it's able to store oxygen when oxygen levels are really low in your body. For example, if you're exercising, oxygen will move from the blood quickly into the muscles because the muscles contain myoglobin, which can bind the oxygen. And this is often found in high concentrations in people who can hold their breath really long because the myoglobin is serving that function. So now that we have seen the different types of proteins, let's take a look at the building blocks of proteins, because that's going to help us understand why there are so many different proteins possible. The amino acid is the monomer of a protein, and the amino acid has a basic structure that's pretty consistent. There's a central carbon, there's an amino group, which is made up of a nitrogen and 2 hydrogens. On the other end, there's a carboxyl group, COOH, and then there's something called an R group. The R group isn't actually the element or the chemical R. The R group can consist of a bunch of different chemicals. So in the amino acids, every single amino acid has the same central carbon, the amino group, and the acid group. That's why they're called amino acids. But different amino acids will have different R groups, and that ultimately is what's going to affect a protein's structure and function. So here we have a polypeptide, and it's composed of 5 different amino acids: valine, histidine, valine again, proline, and threonine. And as you can see, the different amino acids have different R groups attached to them. Histidine has quite a complex R group, where valine has a much simpler R group. So again, it's the R group that distinguishes amino acids. And it's the R groups that are going to give each amino acid a unique property. So if we look at a table, these are the essential amino acids that we'll find making up the proteins in your body. And each particular amino acid has a certain property because of its R group. For example, these 2 amino acids have R groups or side chains that are negative. These 3 amino acids have R groups or side chains that are positive. These have uncharged but polar side groups, and then we've got a bunch of amino acids that have side chains that are nonpolar. So because of these different R groups, The amino acids have various properties. They can also be acidic or basic. So here's an example of how the amino acid properties might actually play out. Here we've got a polypeptide chain, and all of the circles are amino acids. Now the dark circles represent hydrophobic amino acids. Hydrophobic means they hate water. So if you put this polypeptide chain in water, and remember that your cell is mainly composed of water, then the polypeptide is going to fold up like this. It's going to try and keep the hydrophobic amino acids on the inside because they hate water, and it'll keep the hydrophilic amino acids on the outside because they like water. But this is just one small example of how the amino acid R group properties can affect protein shape. Now we're ready to take a look at the whole process. So we're going to close out by looking at the 4 levels of protein structure. The first level is one that we're quite familiar with. This is the primary structure. And as you can see from this diagram, it's simply the sequence of amino acids in a polypeptide chain. But once we have a particular sequence, we can move on to secondary structure. Now this looks pretty complicated, and you do need to draw this in your notes, but please don't draw the individual atoms. What you need to draw in your notes are these 2 shapes. What we have here is an alpha helix and a beta sheet. These are the 2 forms that the polypeptide can take as a result of hydrogen bonds. So here is our original polypeptide chain, but we can see that because there are hydrogen bonds forming between different amino acids, it's starting to coil up into this shape. On the other hand, here we've got hydrogen bonds forming between different amino acids, and it's giving it a pleated shape. So that's our secondary structure. Here's where things get really interesting, tertiary, or the 3rd structure. In the tertiary structure, this is where the R group interactions come into play. So you can see in this folded polypeptide, there's our beta sheets, there's our alpha helices. But now because of R group interactions, those beta sheets and alpha helices are interacting with each other. And this is what's going to give it its 3-dimensional shape. And for some proteins, for simple proteins that are made up of only one polypeptide, this is the end of the line. However, we saw that some proteins, like hemoglobin, are made up of multiple polypeptides. If that's the case, then we have quaternary structure, a 4th level. Here's how quaternary structure works. This is just one fully folded polypeptide, but in the case of hemoglobin, we need 4 of them, 1, 2, 3, 4, to come together to make a complete protein. So quaternary structure is when the multiple polypeptide chains interact with each other. And again, you only find this in complex proteins that have more than one polypeptide chain. Here's another example. We saw that collagen had 3 polypeptide chains. So within the chain, there's its tertiary structure, but then the 3 polypeptides are interacting in this sort of helical fashion to give it a quaternary structure. Similarly, here are the 4 polypeptides of hemoglobin interacting to form a quaternary structure. And that concludes our lesson on protein structure and folding. Please make sure you've got main ideas, vocab diagrams, questions, and themes in your notes. Additionally, I'll post some YouTube videos of protein folding in case you have more questions on how that works. And finally, don't forget to complete the fold.